Hi, I'm Asher Nathan, the CEO of Neotex. First off, I wanted to thank our partner, Active Biotech, and my friends from their management team for allowing me a few minutes to talk about the exciting things that we are doing with the platform technology that we in-license from Active, what we call STR or Selective T-Cell Redirection, and which was formerly called Tumor Targeted Superantigen or TTS by Active. Unlike most immunotherapies, STR is not a band-aid that you put on the immune system to fix one particular problem or another. STR works holistically, like a conductor orchestrating a complete symphony of the immune response. It is not a substitute violin or flute. Rather, STR creates a complete immune response and then unleashes that response onto the tumor. Not only do we believe that STR can turn a cold tumor hot, independent of their etiology, but because it triggers an organic complete immune response, it can synergize with a wide variety of different immunotherapies. Today, I'm going to show you exciting preclinical data showing how STR enables a very effective therapy for blood-based tumors to be effective in solid tumors. As far as our clinical trials go, I will report on our status of our dose escalation trial in combination with CPIs. Currently, we have a collaboration with AstraZeneca and are testing our lead NAP STR-based drug with their anti-PD-L1 therapeutic, as well as a collaboration with Dana-Farber to test the STR platform in GBM. This slide recaps some of the elegant work done by Active in developing this unique platform. The STR molecule is composed of two moieties, third-generation genetically engineered superantigen that achieves an optimized balance between safety and efficacy, and a swappable tumor attachment Fab. Unlike other technology, STR does not lead to nonspecific overactivation of T cells. Superantigen component only triggers T cells from the V-beta 7-9 family, which typically represents less than 3% of a patient's total T cells at baseline. This high selectivity is important both for safety and to avoid overstimulation of beneficial T cells that are present, and it can lead to epitope spreading. Our immune systems are very complex, and there are, of course, lots of moving parts, which means there's a lot of things that can go wrong. Because STR creates a complete immune response, we can cause a productive immune response despite multiple immune system failures. Since the STR recapitulates the entire immune response, what it can do to natural T cells, it can also do to CAR T cells. In addition, one could envision that STR can direct natural T cells to one target and the CAR T cells to another, increasing efficacy and decreasing the probability of resistance. This is from a paper in PNAS from an independent investigator exploring the utility of adding just the superantigen component to CAR T. CAR T can be very effective in blood-based tumors, but it has not had the same success to date in solid tumors. STR is believed to be superior to superantigen for this application for the following reasons. 1. STR is safer than adding wild-type superantigen. The authors themselves point out that it is not feasible to add wild-type superantigen to patients. 2. STR will help in T-cell trafficking and infiltration into the tumor microenvironment. STR will also direct the activation of natural T cells in addition to the CAR T that can also kill the tumor. And 4. STR can direct the natural T cells to a different target than the CAR T, allowing for dual targeting and mitigating the probability of resistance. Here we see the striking results showing only when CAR T was combined with superantigen were complete responders seen, suggesting that STR can enable CAR T to be effective in solid tumors. Okay. I'm sure all of active veteran investors remember this study. We believe that interferon alpha increases anti-drug antibodies substantially and upregulates the PD-L1 axis. To reduce PD-L1, we have partnered with AstraZeneca and will be adding their anti-PD-L1 inhibitor. In addition, we are currently testing ways to decrease or even eliminate anti-drug antibodies in patients in the clinical trial. Currently, we have an ongoing study with NAP in combination with AstraZeneca's anti-PD-L1 therapeutic. The trial is a 3x3 Simon design. The primary goal of the study is to assess the safety of the combination, although we'll be looking for early signs of efficacy. Since NAP has had a history of anti-drug antibody interference, we've initiated an amendment to study the safety and efficacy of a method designed to reduce ADAs. We expect to have the second cycle ADA data from the first 3 patients in the amendment by the end of January. As I mentioned, we have both the ongoing dose expansion as well as the ADA amendment, and will complete both in the first half of 2021. We are planning to initiate the MTD expansion cohort in the first half of 2021, as well as a phase 2 study in lung in combination with docetaxel with a readout in 2023. I want to thank you for your attention and hope to be able to report exciting developments as we progress in our clinical trials. Thank you again.